The next class of organic molecules are proteins. Proteins make up about 20% of our body weight. They are the most abundant of organic molecules in our body. The component elements are carbon, hydrogen, oxygen, and nitrogen. Some proteins also contain sulfur and phosphorus. The building blocks of a protein are amino acids. There are 20 naturally occurring amino acids. The structure of an amino acid consists of 5 components. The first is the central carbon atom. On one side of the carbon atom is a single hydrogen atom. Also attached to the central carbon atom is the amino group, which consists of 1 nitrogen and 2 hydrogen atoms. On the other side is the carboxyl group. This is an acid, which is how we get the name amino acid. The fifth component is the R group. This is a side chain that imparts very specific characteristics to the amino acid. Each amino acid contains a unique R group. Given there are 20 naturally occurring amino acids, there are 20 different R groups, one for each amino acid. Proteins are made by putting amino acids together in a big, long chain. Amino acids are linked together through a process of dehydration synthesis, which we talked about earlier when discussing how to form disaccharides. Water is removed to create a bond between 2 amino acids. The amino acids are linked between the amino group of one amino acid and the carboxylic acid group of the next amino acid. This bond is known as a peptide bond. Peptide bonds are unique to amino acids. Polypeptides, or proteins, consist of a chain of 100 or more amino acids. Proteins are folded and have some type of three-dimensional structure. There are 4 levels of protein structure. The first is the primary structure of a protein. This structure is simply the chain of amino acids. It could be a chain of 100 amino acids or 100,000 amino acids. From this primary structure, we move to the secondary structure. In this case, amino acids begin to form hydrogen bonds with each other. The secondary structure can assume one of 2 forms, either a pleated sheet or an alpha helix. The shape is determined by the hydrogen bonding that occurs between the amino acids in the chain. The tertiary structure creates a more complicated shape to the protein and occurs because some of the amino acids are hydrophobic and must be hidden from the water that surrounds the protein. Additionally, sulfide bonds can be formed, making some of the loops permanent. Lastly is the quaternary structure. This is the most complex and involves 2 or more smaller proteins combining to form a larger protein. We see this in such large proteins as hemoglobin, which is the protein that carries oxygen in your red blood cells. When it comes to protein, as with anything else, shape matters. Remember the structure-function relationship we discussed in the first chapter? If a protein is not properly folded, it will not function properly, causing disease and/or death. Now, let's talk a little bit about the functions of a protein. There are 7 categories of protein function. These include support, movement, transport, buffering, metabolic regulation, coordination and control, and defense. Proteins that provide support include structural proteins that create a 3-dimensional framework for the body. They provide strength, organization, and support for the cells, tissues, and organs. When it comes to movement, contractile proteins bring about muscular contraction. For transport, we have special proteins that bind many substances for transport in the blood, including insoluble lipids, special minerals such as iron, and several hormones. Proteins can also act as a buffer and in this way help prevent changes in the pH of body fluids. Many proteins are enzymes which can regulate metabolic processes. Proteins involved in coordination and control include hormones, which can influence metabolic activities of every cell in the body or affect the function of a specific organ or organ system. Lastly, defense refers to proteins that defend the body in many ways. The tough, waterproofing proteins of the skin, hair, and nails protect the body from environmental hazards. Proteins called antibodies help protect us from disease by taking part in the immune system. Special clotting proteins restrict bleeding after injury. As we go through both Anatomy and Physiology 1 and 2, we will learn more about each of these specific functions in more detail. One of the most important functions of proteins is to function as an enzyme. Enzymes are biological catalysts that are important in catalyzing all the reactions that are used within the cell and required to sustain life. One of the unique features of an enzyme is that it can accelerate chemical reactions without being permanently changed or consumed during that reaction. What this means is that a single enzyme can catalyze the same reaction many times. How do enzymes work? Enzymes contain active sites. Each active site is specific to a specific substrate. Substrates are the targets for the enzyme and are sometimes referred to as the reactants. What happens when an enzyme catalyzes a reaction? The substrates fit into the enzyme like a lock and key to form an enzyme-substrate complex. The enzyme then performs a chemical reaction of some kind, perhaps a dehydration synthesis reaction to remove water, and produces a product that is released from the enzyme. That enzyme is then free to repeat the same reaction using more substrates. Enzymes have three important characteristics. The first is specificity. This means that a single enzyme can only catalyze one type of reaction. The second is the saturation limit. This means the rate of the reaction catalyzed by the enzyme is dependent on the concentration of substrates and the enzyme itself. The third step is regulation, which means that we can regulate that enzyme activity. Regulation allows the cell to turn on or off various enzymes based on need.